I want you to just look down at your hand for a second. Think about the calcium inside those bones or the iron pumping through the veins right beneath your skin. Every single one of those heavy atoms, basically everything other than the hydrogen, was forged by nuclear fusion inside a collapsing star.
It really is wild to think about.
Right. I mean, long before our solar system even existed, the matter that makes up your tissues was being cooked in stellar cores and, supernova explosions. So the chemistry of human physiology quite literally begins in the stars.
It fundamentally changes how you view your own body, doesn't it? Because we usually approach anatomy from the top down. We look at organs, then tissues, then individual cells.
Yeah, the big stuff.
Exactly. But to genuinely understand how life operates, you have to look at the absolute foundation. You really have to understand the chemical rules of attraction that force all those ancient atoms to interact in the first place.
And that is exactly our mission for this deep dive. Today, we are exploring the foundational sources on atoms, bonds, and molecules. We're going to unpack the definitions we still rely on, even though some of them are basically historical fossils at this point.
Yeah, definitely a few fossils in there.
We'll also look at how molecules physically stick together. But more importantly, we want to explore the real-world stakes of this chemistry.
Because this isn't just, abstract theory on a chalkboard. The interactions we are looking at today govern how your nerve cells fire, why ice floats instead of sinking, and this is the heavy one, how the precise 3D shape of a single molecule led to a medical tragedy that forever altered pharmacology.
A huge topic. So let's just start with a piece of language we use every day, which is the word atom. It sounds incredibly definitive, right? It traces back to ancient Greece, to the word atomos, which translates directly to uncuttable.
Right, proposed by guys like Leucippus and Democritus way back in the 5th century BCE.
Yeah, and the original philosophical idea was that if you kept slicing matter smaller and smaller, you'd eventually hit this fundamental indivisible particle. You just couldn't cut it anymore.
Which, it is a beautiful concept, but as a modern chemical definition, it is entirely obsolete.
Totally obsolete.
We still use the word, obviously, but it's a linguistic fossil. Thanks to physics, we've known for well over a century that atoms can absolutely be cut. Under the right conditions, like nuclear fission, they're split right apart.
Right. So today, an atom really just describes the smallest unit of matter that retains an element's specific chemical identity, not the smallest unit of matter in existence.
Exactly. The science sort of caught up in the 1800s with John Dalton's laws, and then Amedeo Avogadro in 1811, who finally distinguished the molecule from the atom.
It is deeply ironic, though, isn't it, that we still use a term meaning uncuttable when particle accelerators are proving it wrong every single day.
It is. But, even after the scientific community accepted the existence of atoms and molecules, there was this massive intellectual hurdle when it came to the molecules inside us.
Oh, right, the vitalism stuff.
Yeah. People were absolutely convinced that human biology was playing by a completely different set of rules. For generations, the prevailing thought was that organic molecules, the ones inside living, breathing things, were fundamentally distinct from inorganic chemistry.
Because they thought there was magic involved, right?
Basically, yeah. The doctrine of vitalism assumed you could never create organic biological matter in a lab because it required this mysterious, intangible life force.
It's like people used to think living things were baked with some magical, unreproducible ingredient. Like, sure, the mountains and the oceans are made of basic chemical flour and sugar, but living things have this magical life spice sprinkled in.
That is a great way to put it. But that whole theory was demolished by a single, really elegant experiment in 1828.
Fried Driller, right?
That's the one. He synthesized urea, which is an organic waste product found in human urine, entirely inside a glass flask. He just used a lifeless, inorganic salt-ammonium cyanate and applied heat. That's it. No living organism was involved at all.
So he essentially baked the biological cake using only rocks and minerals.
He did. And that realization is the bedrock of modern physiology. It proved beyond a shadow of a doubt that there is no separate life chemistry.
We are just chemistry.
Exactly. The molecules that make up your body, your glucose, your hemoglobin, your DNA, they...
By the same chemical rules as everything else, let's explore how those rules actually govern the human body. We know atoms have a nucleus of protons and neutrons, and the number of protons dictates what element we are dealing with.
Right, the protons are the identity.
But it's the variations in the neutrons, the isotopes, that get really interesting from a medical standpoint, according to our sources.
Oh, absolutely. Isotopes are just atoms of the same element that have different masses because of those extra neutrons. And the crazy thing is, human physiology is often completely blind to that mass difference.
Like with iodine.
Perfect example. The human thyroid gland aggressively absorbs iodine from your bloodstream to produce hormones. And it does this regardless of whether the iodine is stable, normal iodine, or a highly radioactive isotope like iodine-131.
Because chemically, the body just sees the electron arrangement of iodine and grabs it. It doesn't check the weight of the nucleus.
Precisely. It's a physiological blind spot, and oncologists actually exploit that blind spot. By administering radioactive iodine-131, they can selectively deliver radiation directly to thyroid cancer cells.
Because the body naturally concentrates the radioactive isotope exactly where the doctors want it to go.
Yeah.
That is brilliant.
It really is. Now, the nucleus determines the element, as you said, but the actual interactions, the bonds, are driven entirely by the electrons orbiting that nucleus.
Right, the valence electrons. From what the sources detail, atoms are essentially on this never-ending quest to stabilize their outermost shell, usually aiming for 8 valence electrons, the famous octet rule.
That quest for stability is the engine of all chemistry. And one strategy atoms use to get there is the ionic bond. This is where one atom completely transfers an electron to another.
Which creates an electrical imbalance, right? The atom that loses an electron becomes positively charged. The one that gains it becomes negative. Opposites attract, and boom, they lock together.
Exactly. The classic textbook example is sodium and chlorine forming sodium chloride, or table salt.
Which is delicious.
It is. But this mechanism is far more than just a culinary trick for your fries.
Yeah.
Your body literally runs on these specific ions. Every single neuron in your brain maintains an electrochemical gradient using a mechanism called the sodium-potassium pump.
The Na plus K plus AT paste pump, right? So by physically shuttling these charged ions across the cell membrane, the body builds up this electrical charge, almost like cocking a spring.
Yes, and when that spring is released, the ions flood back across the membrane. That sudden movement of electrical charge is an action potential.
I think that's a nerve impulse.
Exactly. It is the literal physical mechanism of a thought, a reflex, a heartbeat. And fun fact, we actually figured out this exact ionic mechanism in the 1950s by studying the giant axon of a squid.
Wait, a squid?
Yeah. Hodgkin and Huxley won a Nobel Prize for it in 1963. The squid has nerve fibers so large you can physically insert tiny electrodes right into them to measure the voltage changes.
Oh, wow. That is wild. But I actually want to push back on how ionic bonds are usually taught, though.
Oh, how so?
Well, in a lot of foundational chemistry classes, ionic bonds are described as incredibly strong because of that intense positive-negative electrical attraction.
Right.
But if I take a spoonful of solid ionic table salt and drop it into a glass of plain tap water, it dissolves almost instantly. How can a bond be considered so strong if it just falls apart in a glass of water?
That is a fantastic point, and honestly, a major source of misconception. The strength of a bond is entirely dependent on its environment.
Okay, make that make sense.
So, in a dry, solid state, an ionic bond is remarkably strong. It takes massive amounts of heat to melt dry salt. But human physiology does not happen in a dry vacuum. You are essentially a walking, talking bag of salt water.
Sure.
And water completely changes the physics of the bond. Water molecules are highly effective at forming what we call hydration shells. They physically surround the sodium and chloride ions, insulating their electrical charges from one another.
So inside your bloodstream or your cells,
Yeah.
ionic bonds behave as relatively weak interactions because the watery environment is just constantly prying them apart.
Exactly. And there's actually another major misconception to clear up about salt while we're on the topic.
What's that?
When we look at a grain of table salt, we aren't looking at a pile of individual, isolated sodium chloride molecules. No, and this is where the word molecule can be super misleading. Solid ionic compounds don't form discrete, standalone partnerships. They form an endless, three-dimensional lattice.
Oh, like a grid.
Yes, picture a massive repeating grid of alternating positive and negative ions extending in every direction. There is no single isolated unit you can point to and call one molecule of salt. It is a collective structural lattice until, water rips it apart.
That makes so much sense. Okay, so if ionic bonds are about stealing or donating electrons, the other major strategy is covalent bonding. This is where atoms actually share pairs of electrons to reach that stable 8.
Right, and this is where we get true discrete molecules, and the most vital one for us being water, H2O.
But sharing in a covalent bond isn't always a friendly 50-50 split, is it? The sources mention oxygen is a notoriously greedy atom.
Very greedy. It has high electronegativity, which is just a fancy way of saying it hoards the shared electrons.
So even though the oxygen and the two hydrogens are technically sharing, the electrons spend way more time hanging out near the oxygen atom.
Right, and because electrons are negative, that gives the oxygen end of the water molecule a slight negative charge, and it leaves the hydrogen ends slightly positive. It creates a bent shape, about a 104.5-degree angle.
That unequal sharing makes water a polar molecule. It has poles, much like a magnet.
Yes, and because of those slight charges, water molecules become attracted to each other. The slightly positive hydrogen of one water molecule is drawn to the slightly negative oxygen of its neighbor.
And that creates the hydrogen bond.
Exactly. And this is where the mechanisms of physiology get really, really fascinating. Hydrogen bonds are incredibly weak on their own. They possess only about 5 to 10% of the strength of a true covalent bond.
I like to think of it this way. If a covalent bond is like industrial-grade superglue physically cementing atoms together, hydrogen bonds are basically like Velcro.
Ooh, that's a perfect analogy.
Yeah, because a single tiny hook of Velcro is nothing, right? You can pull it apart with one finger. But if you have millions of them working in tandem, they can hold staggering amounts of weight.
And that collective strength is literally the secret to life on Earth. Water molecules form these vast dynamic networks of hydrogen bonds that are constantly breaking and reforming. And because it takes so much thermal energy to break all these tiny Velcro hooks, water has a massive specific heat.
Meaning it can absorb a ton of heat without its actual temperature rising very much.
Right.
Which is why you can walk outside on a 100-degree day and your blood doesn't literally boil inside your veins.
Exactly. The heat energy from the sun goes into breaking the Velcro of the hydrogen bonds rather than raising the temperature of the water itself. But, the most profound impact of those hydrogen bonds actually happens when water gets cold.
Right, because almost every substance in the universe gets denser as it freezes. The molecules lose energy, they pack tightly together, and it becomes a heavy solid that sinks.
But water completely defies this rule. Ice floats.
And it floats specifically because of the geometry of those hydrogen bonds, right?
Yes. As water cools down and loses kinetic energy, the molecules stop bouncing around so chaotically. This allows the hydrogen bonds to stretch out to their preferred fixed angles. They lock the water molecules into a rigid, crystalline lattice.
But crucially, that lattice forces the molecules further apart than they were in the liquid state. It essentially builds a structure with a lot of empty space inside of it.
Exactly. The solid form is physically less dense than the liquid form. If hydrogen bonds didn't enforce that specific spaced-out geometry, ice would sink.
Which would be catastrophic.
Total disaster.
Yeah.
Lakes and oceans would freeze from the bottom up, and aquatic ecosystems would be crushed and frozen solid every single winter. Life might not even exist.
So a weak transient interaction is responsible for insulating the entire planet's oceans.
Yeah.
That is wild. But the body uses that same weakness for our genetic code, too. DNA relies entirely on hydrogen bonds.
It does. When Watson, Crick, Franklin, and Wilkins finally mapped the double helix structure of DNA in 1953, they realized something amazing. The long vertical backbones of the DNA ladder are held together by that strong covalent superglue.
But the rungs of the ladder, the chemical bases actually connecting the two strands, those are zipped together exclusively by weak hydrogen bonds.
Which is an absolute evolutionary masterpiece when you think about it.
Because if the rungs of your DNA were superglued together with covalent bonds, the cell could never pull them apart to read the genetic instructions or, to replicate them.
Exactly. You need a bond that is stable enough to hold the massive structure together, but weak enough to unzip on demand.
And the specific number of those Velcro hooks matters deeply, too.
It really does. When the base adenine pairs with thymine A to T, they connect using 2 hydrogen bonds. But guanine and cytosine G to C, they connect using 3.
So a section of your genome that happens to have a lot of GC pairs is physically stitched together more tightly.
Right, it has a higher thermal stability. The cellular machinery actually requires more energy to physically unzip a GC-rich region of DNA than an AT-rich region. So the physical sequence of your genetics directly dictates its mechanical strength.
That is incredible. Now, we've been contrasting the weak Velcro of hydrogen bonds with the strong superglue of covalent bonds. If we want to see that superglue at work in human physiology, we really just have to look at ATP.
Adenosine triphosphate, the energy currency of the human body.
Right. ATP stores massive amounts of potential energy in the covalent bonds connecting its phosphate groups, specifically those phosphoanhydride bonds. When a muscle needs to contract or a cell needs to pump an ion, an enzyme physically breaks that terminal phosphate bond.
And hydrolyzing that bond releases a highly specific amount of explosive energy, about 7.3 kilocalories per mole.
That single chemical snap is what powers nearly every active biological process keeping you alive right now.
Which actually brings us to a conceptual leap that is incredibly difficult to visualize, but absolutely necessary if you want to understand this stuff.
Okay, lay it on us.
Well, we learn about bonds using those little plastic ball and stick models in high school classrooms, right?
Right, the little molecular model kits.
Yeah. And we are taught to view molecules as this rigid, static architecture. But inside your body at 98.6 degrees Fahrenheit, molecules are in constant, violent motion.
They are vibrating, flexing, bending all over the place.
Exactly. A bond is not a rigid piece of plastic. It acts a lot more like a stiff spring. And this molecular flexibility is a strict requirement for physiology.
How so?
Well, when an enzyme in your body needs to bind to a target, they rarely fit together perfectly like a rigid lock and key. The enzyme and the target literally flex and mold around each other in what we call an induced fit.
Oh, so the 3D shape and the ability to change that shape directly dictates function.
Yes. And a molecule's geometry is so critical that a microscopic shift in its 3D arrangement can literally be the difference between a life-saving therapy and a lethal poison.
And there is no darker example of this than the history of thalidomide, which the sources highlight in detail. In the late 1950s and early 60s, thalidomide was marketed by Chemie Grunenthal as this gentle sedative and a highly effective anti-nausea medication.
Right. It was widely prescribed globally to pregnant women to combat morning sickness.
But the chemical structure of the drug hid a devastating geometric secret.
It did. Thalidomide is what we call a chiral molecule, meaning it exists as two different enantiomers.
Enantiomers?
Yeah, enantiomers are basically mirror images of each other. The easiest way to visualize this is to just look at your own hands. Your left hand and your right hand are made of the exact same components, 4 fingers, 1 thumb.
Yeah.
But no matter how you turn them, you cannot perfectly superimpose your left hand over your right hand. They are mirrored.
So these two versions of thalidomide share the exact same atoms, connected by the exact same covalent bonds, but they are permanently mirrored in 3D space.
Exactly. And in the highly specific, very sensitive chemical environment of a developing fetus, that mirrored geometry was catastrophic. One enantiomer of thalidomide functioned perfectly as a safe sedative, but its mirror image was intensely teratogenic.
Meaning it disrupts fetal development.
Right. Wedukin Lenz and others documented that it interfered with the development of blood vessels in fetal limbs, causing severe malformations.
Over 10,000 children were born with profound physical defects before the drug was finally pulled from the market. But you know what I found truly terrifying in the research?
What's that?
It's a detail about how the body actually handles chirality. Let's say a pharmaceutical company back then figured out how to filter the drug perfectly, and they gave a patient a completely pure dose of only the safe sedative enantiomer.
Oh, yeah. It wouldn't have mattered. It would not have prevented the tragedy.
Because the human body itself alters the geometry.
Yes. The dynamic chemical environment of the human bloodstream, the pH, the temperature, the enzymes, it actually causes the thalidomide molecule to undergo spontaneous interconversion.
So the bonds briefly break and reform, literally twisting the safe molecule into its lethal mirror image inside the patient's own body.
Exactly. You simply cannot isolate a chemical from the dynamic environment you put it into. And that tragedy forced regulatory agencies worldwide to completely rewrite how drugs are tested.
I can imagine.
Today, scientists must rigorously map the 3D shape of a molecule and test every possible enantiomer, specifically because the body's machinery is so incredibly sensitive to spatial geometry.
Well, if the precise 3D space a molecule occupies is quite literally a matter of life and death, it raises a really frustrating problem.
Yeah.
How do we even visually map a chemical bond? Because a bond isn't a physical wire we can just look at under a microscope.
No, it's not. It is a mathematical region. A bond is simply an area of concentrated electron density between two nuclei, governed entirely by the bizarre, counterintuitive rules of quantum mechanics.
And since nobody can easily visualize quantum mechanics, chemists had to invent visual shortcuts. That's where Gilbert N. Lewis and valence bond theory come in, giving us those famous Lewis dot structures back in 1916.
Oh, the dot structures. We've all drawn them.
Right. You write the chemical letters
Yeah.
and pair up the little dots between them to represent the shared electrons.
And look, Lewis structures are brilliantly intuitive. They treat electron pairs as heavily localized, like they are trapped directly between two specific atoms. And for basic organic chemistry, they work wonderfully.
Hmm.
But when you try to apply that simple localized model to certain molecules, the model fails spectacularly in the real world.
And the ultimate example of this failure is the very oxygen you're breathing right now.
Yep, O2.
If you sit down and draw a Lewis dot structure for O2, all the electrons pair up perfectly in a neat little double bond. And based on that drawing, oxygen gas should have absolutely no magnetic properties.
Because magnetism at the molecular level is driven by unpaired electrons spinning in the same direction. If all the electrons are paired up, as the Lewis structure suggests, their spins cancel each other out. No magnetism.
But if you go into a lab and you take liquid oxygen, which is just supercooled O2, and pour it between the poles of a powerful magnet, the liquid oxygen will defy gravity and physically cling to the magnet.
It gets trapped right in the magnetic field,
Yes, liquid oxygen is highly paramagnetic.
which is physical proof that oxygen must have unpaired electrons. The simplified dot structure is blinding us to reality. To understand why oxygen is magnetic, chemists like Mulliken and Hund in the 1920s had to develop molecular orbital theory.
Which forces us to abandon the idea that electrons just belong to individual atoms.
Exactly. So instead of trapping the electrons on a little line between two atoms, molecular orbital theory shows that the electrons occupy these vast, complex orbitals that spread across the entire molecule.
And when you map the math of those molecule-wide orbitals, it correctly predicts that oxygen has two solitary electrons sitting in completely separate regions of the molecule spinning independently.
And those two unpaired electrons generate a microscopic magnetic field.
It kind of makes you wonder if we should even be teaching the simple dot structures if they lie about something as fundamental as oxygen.
Well, I wouldn't say they're lies. They aren't lies. They are just different approximations.
Different tools for different jobs.
Exactly. A biologist mapping out a simple sugar chain doesn't need quantum orbital math. A Lewis structure is the perfect tool for that. But a biochemist trying to figure out how a specific protein binds to oxygen, they absolutely need the deeper quantum model.
And speaking of binding to oxygen, that is exactly the problem human physiology had to solve. How does the body capture this weird paramagnetic molecule and transport it?
The solution is hemoglobin. Max Perutz mapped this out around 1959-1960. It is one of the most mechanically elegant proteins in nature.
How does it work?
Inside hemoglobin are 4 heme groups, and each one contains an iron ion. That iron forms what is called a coordinate covalent bond with an oxygen molecule in the lungs.
But it's not just a static docking station, right? This brings us right back to the idea of molecules flexing and moving.
Yes, exactly. When that first oxygen molecule binds to the iron, it physically tugs on the iron atom, pulling it slightly out of position.
Just a microscopic pull.
Right, but that microscopic physical pull triggers a massive cascade. The movement of the iron atom warps the entire massive 3D structure of the hemoglobin protein. As it bends, it exposes the other 3 iron binding sites, making it dramatically easier for the next 3 oxygen molecules to snap into place.
Ah, it's called cooperative binding.
You got it.
So the hemoglobin physically changes its shape to become more aggressively magnetic to oxygen as it passes through the lungs, and then flexes back to drop the oxygen off deep in your tissues where you need it.
It is breathtaking engineering, and it perfectly encapsulates everything we've explored today, from the specific isotopes of iron to the complex geometry of the protein to the induced quantum fit of the bond itself.
Which is really the ultimate takeaway here. We've covered Greek philosophy, quantum mechanics, and, squid biology. But none of this is isolated trivia. This specific chemistry is the operational manual for the world around you. When you grab a sports drink, you are actively managing your ionic bonds, ensuring your sodium-potassium pumps have the ammunition they need to fire your nervous system.
And when a hospital runs a PT scan to detect a tumor, they're relying on radioactive isotopes like fluorine-18, seamlessly integrating into the body's natural covalent structures to map metabolic activity in real time.
And let's not forget the recent leaps in mRNA vaccine technology. Those are a masterclass in this chemistry. Those vaccines only function because scientists have achieved near-perfect control over the rigid covalent bonds in the molecule's backbone, while carefully preserving those fragile, Velcro-like hydrogen bonds needed for the cell to actually read the genetic sequence.
It's all connected. Every thought you have, every heartbeat, every immune response is, at the most fundamental level, just millions of bonds breaking and reforming in perfect synchronization.
Which leaves us with a genuinely profound thought to mull over. We started this deep dive by acknowledging that the atoms inside you were forged in ancient stars.
Right.
But under normal chemical conditions on Earth, matter cannot be created or destroyed.
It can only be rearranged.
Exactly. That means the very atoms currently bonded together to form your heart, your brain, and your blood have been endlessly recycled since this planet coalesced.
Just endless combinations over billions of years.
The carbon and oxygen molecules that make you who you are today were once part of prehistoric oceans, primordial forests, and entirely different living creatures. Right now, in this exact moment, they are temporarily bonded together in a fleeting, beautifully complex configuration, just waiting for their next chemical reaction.
A true masterpiece of temporary chemistry.
It really is. Thanks for joining us on this deep dive. We'll catch you next time.